Introduction, Sean. And thank you for giving me this opportunity to present this talk. I'm Anusri Pampari. I'm a computer science PhD student at the Cooperative lab. Today, I'm about to present our work on Chrome BPNet models, which are deep learning models of base resolution chromatin profiles that reveal cis-regulatory syntax and regulatory genetic variation. I will just try to point out. Yeah, no, good. As you all know, transcription factors cooperate with each other and compete with nucleosomes to get access to DNA and bind specific sequence domains. So regulatory elements bound by TFs are associated with accessible chromatin, as well as a variety of histone modifications that encode for combinatorial chromatin states. So deciphering the DNA sequence basis of TF binding and accessibility is an important step to understanding gene regulation. A popular approach to profiling regulatory DNA relies on chromatin immunoprecipitation methods such as ChIP-seq. However, ChIP-seq methods require high input cell numbers, are limited to 1 TF per assay, and are restricted to TFs for which antibodies are readily available. Therefore, it remains costly or even impossible to study the binding of multiple TFs in parallel. So chromatin profiling experiments like ATAC-seq and DNA-seq provide a promising alternative and allow for systematic global investigation of regulatory DNA. To date, all machine learning approaches such as SVMs and neural networks that learn to map DNA sequence to chromatin profiles do not model these data at single base resolution. The signal coverage is typically binarized based on peak calls, or the signal is aggregated into lower resolution bins. So these models miss the complex, multi-resolution shapes and spans exhibited by these profiles. Further, at single base resolution, chromatin profiles reveal footprints that are characteristic of both TF binding and accessibility. These footprints are hard to uncover at lower resolution and without accounting for enzyme preference bias. So the enzymes used in these assays, such as TN5 and ATAC-seq and DNase1 and DNase-seq, exhibit distinct sequence preferences, such as the PWMs shown here, so they can bias the profile significantly from the true accessibility profile. Existing methods like hint attack, 2-bias, Olher et al. Have looked into PWM and Kramer-based models to characterize the bias and correct for it. However, in our work, we see that these simple models do not adequately characterize and correct for it, and hence enzymatic bias continues to be an important confounder for existing predictive sequence models like neural networks. So to address both these limitations, we introduced Chrome BPNet, the first end-to-end deep learning models that learn to map DNA sequence to base resolution chromatin profiles while accounting for assay bias correction. So these models are fully convolutional neural networks that take a 2 kb sequence as input and predict 1 kb output in two complementary quantities. The first is the total TN5 DNase insertions in the 1 kb region, that is the total signal. And the second is the base resolution probability profiles, which is the total shape. We account for assay bias correction in 3 steps. We use a simple neural network to learn to predict at base resolution the background regions, excluding the peak regions, from the underlying sequence. The redistribution in the background regions is a function of enzyme bias. So these models will capture the sequence preference bias, as I show here by the TN5 motif captured by the model trained in ATAC-seq background. Further, these neural network models are powerful, and hence they can capture a richer and a heterogeneous representation of the TN5 motif compared to the simple PWM representation of TN5 that I showed you in the previous slide. Next, we use these models to predict the bias in the peak regions and to fit the measured ATAC-seq profile in the peak regions to the predicted bias profile. Finally, we freeze the bias component of model and fit the sequence component of the model to the residual of the ATAC-seq DNase-seq signal that cannot be explained by the bias. So this multi-step regression approach ensures that the sequence component of the model does not learn enzymatic bias. So to summarize the performance of Chrome BPNet models, I show you here a performance of a model trained in K562 DNase-seq and its prediction performance in peak regions and held out test chromosomes. These models achieve a high Spearman correlation of 0.7 between the observed and the predicted total counts. We also compared the observed and the predicted base resolution probability tracks by using junction Shannon distance or JST as a metric. We use the JST between profiles of pseudoreplicates as the best limit, and the JST between observed and the shuffled, and observed and the mean summit centered as the two worst case limits. Our models are significantly better than the worst case limit and approach the pseudoreplicate concordance. Now, let's look at chrome-BPNet's bias correction in action. Consider here an example locus in GM12878 ataxic signal in chromosome 20, and consider using the chrome-BPNet model, but without the bias correction. Here, we show that the observed track is very similar to the predicted track when bias correction is not applied. When we try to use the deep lift algorithm to interrogate the model and derive contribution scores at individual sequence nucleotides that are predictive of this profile predicted, we not only see high contribution scores at a CTCF motif, but also noisy with strong contribution scores at spurious nucleotides. We note that this closely matches the TN5 motif. Let's look at how chrome-BPNet performs. If you look at the bias model trained in the background regions and use it to predict the bias profile in the peak regions, it predicts a profile which is remarkably similar to the observed track. And when you try to derive contribution scores to explain this model, you see that it highlights bases that closely match the spurious nucleotides in the uncorrected model. So this shows that TN5 bias has a profound influence on the ataxic profiles even in the peak regions. On the other hand, the bias-corrected profiles from the chrome-BPNet model shows a dramatic correction, clearly showing the footprint bookending the CTCF motif. And the contribution scores are totally devoid of the spurious nucleotides, clearly highlighting the CTCF motif. So chrome-BPNet models are able to perform extremely effective bias correction while improving the quality and resolution of footprints at individual sequence elements with sparse coverage. Comparing this to existing methods like hint attack and 2-bias, we show that these models do not adequately correct for the bias and do not reveal the smooth footprint of CTCF. Here, we compared the chrome-BPNet models trained in two different chromatin profiles, ataxic and DNE-seq, and in the same biosample, KPA62. We show high concordance between bias-corrected predictions across the two profiles compared to observed and the predictions without bias correction. More so, the contribution scores after bias correction show remarkably high concordance, showing that chrome-BPNet models are able to perform extremely effective bias correction and reveal regulatory syntax that is consistent across both the assays. Now, having achieved high performance, we turn our attention into deriving the predictive sequence features which are responsible for our accurate predictions across different cell types and across different chromatin profiles. We start with using the TF-Modisco algorithm on our deep lift contribution scores to get consolidated motifs. I'm about to show you the 9 most prevalent predictive motifs identified by the TF-Modisco algorithm from our chrome-BPNet models trained in ataxic in 4 different cell lines. We see expected cell type specific motifs appear such as IRF, RunX, SPY1, NF-kappa B in GM12878, GATA in K562, FOXA, HNF4J in HG2, OCT-SOX, ZIG3, SOX2, and TED in H1HACs. TF-Modisco also identifies other non-cell type specific motifs. We show that chrome-BPNet models are able to accurately account for the bias and denoise the signal of its enzyme effect, revealing footprints at cell type specific TFs. So here we embed these motifs in 10K background sequences and use each of these cell type specific models to make both uncorrected and corrected predictions on these synthetic sequences with motif insertions. Now for every motif, we average the predictions across all the synthetic sequences to get these marginal footprints. We show you here that the GM1287 ataxic corrected model shows the footprint only for NF-kappa B, but not GATA and HNF4A. Similarly, the K562 corrected model shows a footprint only in GATA, and HEPG2 corrected model shows a footprint only in HNF4A. SP1 is a non-cell type specific TF and hence shows a footprint for all three models. We also know that the footprints identified by chromBPNet DNA-seq models are much finer in detail and are more compact compared to the footprints of the ATAC-seq model. This allows us to capture much deeper footprints in DNA-seq compared to ATAC-seq, as we show here using NFRB. So these differences are consistent with the differences in the enzymes. For example, TN5 is much larger than DNase1 and hence induces broader footprints. We can also use chromBPNet models to study motif-mediated cooperative effects of TFs on the chromatin profiles. Here, I'm showing you an example of an AP1 plus TED heterodimer that is predicted to produce a superadditive profile compared to only AP1 and TED motif, TED TF only. Now, let's use chromBPNet models for regulatory variant effect prediction. I'll start with a case study of a variant that was previously identified as a binding QTL and spivin transcription factors in LCLs like GM12878. We will use three models to query the effect of the variant on three different regulatory readouts. The first is a model trained in spivin TF chip-seq. The second is a model trained in DNA-seq, and the third is a model trained in H3K27AC chip-seq. We first predict these three profiles by introducing the C allele in the sequence surrounding the variant. We see moderate signals for all three models. Next, we introduce the G allele in the sequence surrounding the variant and again use the three models to predict binding, accessibility, and acetylation. We see a dramatic amplification in the signal for all the three models. This observation is consistent with this variant being previously identified as a strong and a statistically significant BQTL. We can then compare the profiles of both the reference and the alternate allele to reveal the blast radius of the variant, which is the range of propagation of the differential signal. Finally, we contrast the deep lift scores of both the reference and the alternate allele and see that there is a weak spivin motif which is driving the signal in the reference allele. But the alternate allele creates a strong spivin motif directly overlapping the variant, resulting in the dramatic amplification that we were seeing before. We systematically study this by using chromBPNet models trained in GM1287 ATAC-seq to predict spivin BQTLs. We show here that these models are able to predict effect size and direction of effect with a high Spearman error of 0.64. We also evaluate the ability of chromBPNet models, the variant effect scores from chromBPNet models, to distinguish between DNase QTLs from corresponding matched control variants. Here we use a GM1287 ATAC-seq model for this. We show that our models outperform existing methods like delta SVM in predicting DNase QTLs. So to summarize, I showcase chromBPNet models, which are accurate base resolution profile models for ATAC-seq and DNA-seq experiments. These models provide superior enzyme bias correction and can provide denoise footprints at base resolutions at individual enhancers. By using deep lift and TF modisco, we reveal predictive motifs and motif instances and can also reveal cooperative effects of syntax. Finally, I also showcase that chromBPNet models are able to predict effect size and direction of effect of both binding QTLs and DNase QTLs. We hope that chromBPNet models will be another powerful tool to dissect the cis-regulatory syntax of the chromatin code. With that, I would like to thank Anna Sharabina for her Cell Plus partnership, Surag, Avanti, Amin, Austin, and Shomo for their help with some of the analysis, and Anshul for his guidance and support. We would also like to thank our funding sources. Thank you for listening. Open to questions and feedback.
Yeah, thank you for this cool talk. So you said, the bias correction is better than hint and bias, but you didn't really show any result when you elaborate on this. So I think it makes sense, but I'm also wondering, in hint and we've seen that it's important, because it's a dimer, hint 5, that you basically model this, and then also the fragment size of the where cuts basically plays a role. Is this all learned by the by the network for the bias? Did you look at this?
Yeah. So to go back to the result, so this model here uses two bias correction. So we evaluated extensively on how well two bias corrects, and we show that they clearly like show a TN5 motif. Even if you once you train a model on the two bias corrected tracks, and then if you interrogate it, you actually still see TN5 motif. So the reason I think why these existing models were not able to figure this out was because they never had the contribution scores to see if the TN5 bias was fully corrected or not. Yeah, what was your other question? Sorry.
Whether you looked into if your network learns this, basically the symmetry of the TN5 bias,
Yeah.
The fragment length makes a difference.
Yeah, it does learn the symmetry of the TN5 bias. In fact, it learns like a much richer representation of the TN5 motif. Like when you use PWMs, you're like summarizing the entire different representation of the TN5 motif that you can see. And I had a slide. Yeah, so this is the original TN5 motif that was used by two bias and hint attack. And these are the other representation of the TN5 motif that are actually captured by the model.
But in hint attack, we don't use PWM, right? It's a dependency model where you have very order memory and you model dependencies. But I mean, I believe that it's better. It makes a lot of sense.
No, hint attack uses a Kramer before. Like you use a set of sequences which are of a fixed length, and then you try to use a similarity score between them.
I don't want to.
Oh yeah, sure. We can discuss it later for sure.
I was just curious whether you learned this.
Yeah. Thank you.
Thanks, great talk. And I was wondering about the fact that you actually can capture better footprints with DNA in comparison to attack. And you mentioned that these are the transcript TM5 motifs, and I was wondering why your model wouldn't be able to learn longer sequences of TM5 motif in comparison to DNA.
The motifs are longer. Like these are the full motifs that are captured by TN5 for the TN5 motif. And if you look at the motifs which are captured for DNAs, they are much shorter in length. I don't have a slide for that, but they are definitely much shorter and compact.
And then why it is harder for your model to learn?
No, it is learning both the motifs. Like if you train the neural network on the background of the ataxic data, you learn the TN5 motif. And this is only a model which is trained on the background of attack. But if you train the same model in the background of DNA-seq, then you pick up the DNase1 motifs, which are much smaller in size.
Yeah, so what is then the better in DNA-seq in comparison to ataxic? Why do you capture something better in DNA-seq in comparison to ataxic? And sorry, I think I missed that question.
No, both of them are equal. It's not we do the bias correction equally in both. TN5 is a much harder bias to correct for than DNase-seq because it has a much stronger influence. So it's kind of like a validation that once you have corrected, you're actually having similar predictions to DNase-seq as well. So your footprints and everything look very similar. So if you look at, where is that slide? If you compare the ataxic and DNase-seq profiles, you see that the observed track if you just look at the observed track in ataxic and DNase-seq, they don't look similar at all. They are the same, they're sequencing the same chromatin profile, right? But they don't show footprints at all in the ataxic. But after correction, you start seeing footprints for both DNase-seq and ataxic. So it's just that it's much stronger, so it biases the profile a lot more from its true accessibility profile. So it's much harder to correct. And these models are able to do both the corrections properly.
Thank you.
Just one brief question from online here. Max Liebert is asking, could you go into more detail on the model architecture? Are the bias and bias corrected signals two outputs of the same model, or are there two disjoint models? And then also he asks, have you looked at how important it is to include the bias as an output with deep lift interpretation?
I'll answer the first question. Can you repeat the second question later?
Yes.
So there are two disjoint models. So we kind of train a bias model, then you freeze it, and then train a sequence model on the residual signal that cannot be captured by the bias. So this shows that the residual signal does not include bias. So the second model that you're actually learning will not have any bias included in it. So there are actually two disjoint models that are trained together. Yeah.
And then the second part was, have you looked at how important it is to include the bias as an output for the deep lift interpretation? You definitely showed individual examples, but have you globally?
Yeah, it is definitely important to, I think it's important to verify that your background models are not capturing any motives, because you don't want to regress the effect out. So it's important to make sure that you see that in your contribution scores, that you only capture T and phi or the Dini's one motives or any enzyme motives, and not TF specific motives.